It's my pleasure to introduce Dr. Hartwell. He earned his BS at the California Institute of Technology and his PhD from MIT. He won the Nobel Prize in Medicine and Physiology in 2001. He won that prize for looking at the regulation of yeast genomics and cell duplication, which has been determined to be fundamental to understanding cancer. So it really is my pleasure to welcome Dr. Hartwell.
Thank you very much, and it's also a great pleasure for me to be here. How do we deal with the more general population, the average person who is at risk for cancer? And how do we deal with other diseases besides cancer in going in this direction of individualized and more preventative medicine? And I'm going to be stretching things a little bit. I'm going to be looking into the future rather than the present, but I don't think it's too far into the future. I've been a basic scientist for about 40 years studying cells and cancer, and over that time, the field has learned a great deal about this disease that we didn't know 40 years ago. What we've learned is that about a half a dozen different cellular processes become altered in a cancer cell. And it takes a lot of changes to convert a cancer cell to a normal cell. That's why, even though we have so many cells in our bodies, cancer is relatively rare and a phenomenon mostly of older age. And these are some of the processes that get changed. For example, the regulation of the growth of the cell, where normal cells depend upon other cells to stimulate them. Cancer cells become self-sufficient. Normal cells are mortal. They only go through a certain number of divisions. Cancer cells become immortal. Resistance to cell death, angiogenesis, they grow blood vessels to them. Metastasis, they migrate in the body, whereas normal cells don't. And this area of the cell cycle, which alterations lead to genetic instability. And that's a really fundamental thing about cancer, is genetic instability. So cancer, unlike other diseases, is motivated by genetic changes in the cell that we can detect and use as diagnostic markers for what's happening. And it's a whole area of diagnosis that I really want to emphasize. Now, this is a graph showing the mortality, age-adjusted mortality for different diseases, the major diseases, heart disease and stroke. And heart disease is in green, stroke is in purple. And you can see since 1950, both heart disease and stroke have decreased by a factor of about 3. Considerable progress. We don't know why, by the way. The interventions that are popular for these diseases, like statins, for example, didn't come in until the mid-'80s. But nevertheless, progress due to some reason. But cancer is the yellow bars, and there's been very little progress in the area of cancer. We can point to some dramatic improvements in some relatively rare cancers, but for the most part, it's not a very encouraging story. However, the most encouraging thing about cancer is this story here. This happens to be colorectal cancer, but it's true for most other cancers. Look just at the last set of 3 bars there, 1993 to 1997. The blue bar represents survival, 5-year survival, for people who were detected with localized disease. And the maroon bar represents people who were detected with what's called regional disease. So there's been a little bit of spread, but it hasn't gone very far. And the white bars at the bottom, or yellow bars, are for people with metastatic disease. And what's very dramatic for almost all cancers is that survival is very good with current treatment. We don't need a new treatment for cancer if we detect it early. For the most part, surgery and radiation will cure cancer at an early stage. And what's also dramatic is if you look backwards from 1973 to 1993, the story hasn't changed very much at all. But you can see a slight improvement in outcomes, both for the blue bar and for the maroon bar, and even maybe a little bit for the yellow bar. And that's our degree of progress. But clearly, if we could detect more cancers earlier, we would cure a lot more people. This is just the stage at which people present, the proportion of cancer patients presenting with local, regional, or distant disease at these different time intervals. And you see that really hasn't changed very much either. And you'll see a similar story with each type of cancer. So the thing that I'm very interested in is, can we find diagnostic ways to detect cancer early? And you heard about some of the methods now which depend upon imaging technology for the most part, looking at things. Colonoscopy we can detect early because we can put a fiber optic up the colon and look and see if there's disease or not. And mammography is a way of looking too. And Norm Beauchamp, who will talk after me, will talk a lot more about those imaging technologies. But what I want to do is raise the question of whether we can use molecular markers to detect disease in a less invasive and, more importantly, a less costly way, because colonoscopy is also about $1,000, and one reason it's not more popular is because of the cost. There are other reasons too. These are the questions that we would like to be able to answer for ourselves. Am I likely to get cancer? That's the risk question, okay? Do I have cancer? And if I have cancer, what stage is my cancer? So this is the detection and particularly the interest in detecting cancer at an early stage. And is my cancer responsive to therapy? Some cancers are, some cancers aren't. And if it looks like a type that's responsive to therapy and I'm being treated, is the therapy actually working? And being able to answer these questions really will begin to move us from an area where everybody is treated the same to this era of individualized medicine, which is not very far on the horizon. So because cancer is a genetic disease and there are DNA changes in the cells, unlike most other diseases, we can use those DNA changes to answer some of these questions. The question about risk, families at risk, where we actually know some of the mutations and can test individuals to see if they have that mutation long before they have cancer. As I say, that applies to only a small percentage of people at risk, but nevertheless, it's an example where we can, using molecules, diagnose the risk and assign a very reliable sort of measure to that. The question of do I have cancer or not is a detection question, and a particular important study that's going on now with esophageal cancer, which if detected late is essentially as deadly as pancreatic cancer, is a study conducted by Brian Reed here at the Seattle Cancer Care Alliance, where they have identified some patients who are at risk because of continuous acid reflux into their esophagus, and they see these patients every year or so, and they take small samples of the esophagus and look for DNA changes. And you can detect certain DNA changes which are indicative of a high probability of progression to cancer and actually early stage cancer. And the survival for those patients where surgical intervention occurs early is quite high, 80–85% or so over the course of the study, compared to what would be 5% or less 5-year survival if it was just detected when symptoms arose. Another example is, for example, for the prognosis of whether the particular cancer is responsive to therapy. There are many examples, but one is chronic myelogenous leukemia, which has a specific DNA rearrangement. It's a kind of leukemia that's diagnosed by a very specific DNA rearrangement. And if that rearrangement exists, then there's a drug that's very, very highly effective. In fact, it's the best drug we have in cancer. It's Gleevec. You've probably heard of it. But it puts people into complete remission if they have that disease. And there's only about a 4% per year progression for people on that drug. So that marker tells you these people will respond to that particular therapy. And then, is the cancer actually responding to the therapy? In that particular case, again, you can just take samples of blood from the people, and if they're responding to the therapy, then the frequency of this particular DNA marker in their blood drops dramatically, 1,000-fold. And you just follow them over the course of their treatment. And if it starts back up again, that's an early recurrence of disease, and then the only treatment that's effective at that time is a bone marrow transplantation. But if you don't detect it early, even a bone marrow transplantation won't work. So being able to follow this DNA marker, not only for the response to the treatment, but for the recurrence of the disease, is critical in managing this particular disease. This is one dramatic example of using DNA markers in cancer. This is for infants less than a year old for survival to neuroblastoma. And those children who have a particular DNA amplification have a very, very dangerous disease with high mortality for which there's no cure. And you can see their survival is very, very low, most of them dying within 2 years. Whereas if they don't have that particular DNA change, then almost all of them survive. So we're in this era in cancer when, because there are DNA changes in all cancers, we're not just talking about those inherited DNA changes which lead a small percentage of people to be highly susceptible, but we're talking about any of us who get cancer. There are DNA changes in those cells that can be monitored and can be very, very highly effective diagnostic markers. Now, the problem is that most of the diseases that we're interested in besides cancer don't have DNA changes to look at that way. And for many of the things, even for cancer that we would like to detect, we can't go to the cancer site and monitor. I mean, before a person has disease, if you want to know if they're at risk for disease, there's no site to sample. If you want to know whether they have early stage disease before there's a lump or you can see it, there's nothing to sample, right? And often, most times with response to therapy, there's nothing to sample because you've taken it out, you're giving them chemotherapy, and there's nothing to follow. So DNA markers, I think, are just pointing to the future of molecular diagnostics that will become much more powerful when we can use proteins as better diagnostic agents. Now, as you know, the genes are made of DNA, and genes code for proteins, which then go out and perform the functions in the cell. And proteins are actually much more informative than DNA for 2 reasons. One is they're much more diverse. So because of a variety of things that happen along the way from making from a DNA molecule to making an active protein molecule, there's a lot of diversity at the protein level that's not present at the DNA level. So it's estimated that a single gene can produce at least 10 different forms of a protein, and probably many more than that. There are at least 100 chemical modifications that can occur to proteins after they're made that affect their function. So proteins are much more diverse and have much more information. They're likely to be much more accessible because they're likely to be circulating in body fluids. And also, they're much more indicative of what's going on at the functional level, because proteins do the function. Okay? So they're really informing us of the functional state of the cell. And so, I think the future is to be able to diagnose from the proteins that are present in a person what their physiological state is, and to be able to answer all these questions about risk and early stage disease and whether a person's responding to therapy and all of these things. The limitation at the present time is that the technology for measuring proteins in the kinds of diversity that we will need to is still in its infancy. The technology for measuring DNA is very, very sophisticated. We can measure a single molecule of DNA. We can take a single cell and amplify all its genes and measure the messengers from those genes from a single cell. Very, very sophisticated technology. But the protein technology is just beginning, but I think it's very, very likely that that information will be there. And the reason is, of course, because we have a number of protein markers already that we use routinely for diagnosing physiological states. A test for pregnancy is a test for a protein that's pretty reliable. Test for heart infarction is proteins troponins, which circulate in the blood after a heart attack. Medullary thyroid cancer, a rare form of cancer, it's actually the best cancer we have a protein marker for, calcitonin. So for some things we have discovered it, and what's encouraging is that in the last couple of years there's been a real advance in the technology for accessing, identifying, and quantitating proteins from complex mixtures of proteins like blood. And an essential component of that is the human genome information. That is, to be able to take a peak that appears in a mass spectrometer and say what protein that is requires a lookup table. That's the catalog of all of the human proteins that we get from translating the DNA sequence. So the completion of the human genome just a few years ago has given us a capability that we didn't have just a few years ago. So I think we're going to see a big advance in protein diagnostics as a result of that. This is just an indication, though, of the difficulty that we have, which is this is a graph of a number of proteins listed along the body here at the bottom from that are known proteins present in serum, and on the vertical scale is their concentration in blood. And if we were talking about DNA, then all of the genes would have the same concentration. There's 1 or 2 per every cell. But when you look at proteins, this is a log scale. And there's a 10 to the 11th fold range for the very minor proteins compared to the abundant proteins. 11 factors of 10, okay? Huge difference. And so the technology currently can access the most abundant proteins, but is not very good at accessing these very minor proteins down here where the real diagnostic information is. Down in this area of the green and yellow is where proteins that come from tissue leakage, and the red ones are ones which are known to be highly functional proteins in the blood. And so what's necessary is to apply the technology that exists in novel ways, and I equate this sort of to a human genome kind of project that requires a rather large-scale activity of a lot of technology aspects with data standards and reagents and everything. But I think it's really possible over the next 5 years or so to really crack this area of protein diagnostics and be able to answer many of these questions about disease, not only for cancer, but for many other diseases. And as we do that, we'll get into this area of individualized medicine. We already are finding that with respect to chemotherapeutic treatments and immunosuppressive treatments, for example, for bone marrow transplantation, that different individuals need different levels of treatment. And we can give them tests and decide what actual level to treat them. And this whole area of molecular diagnostics will lead us into that area of much, much more individualized tuning of therapeutics. So I think that's all I have to say. Thank you.
Thanks, Lee. Well, congratulations. You've made it to your final lecture of mini med school. The topic for your final lecture will be anatomic and molecular imaging. There is a tremendous alignment between molecular diagnostics, individualized therapy, and imaging. The alignment comes about because of the shared goal of lessening suffering, disability, and death related to cancer. As you're hearing, it's really a multidisciplinary approach with expertise from across the field of medicine and with a number of different groups, genomics, bioengineering, imaging, and the imaging sciences contributing. Given the alignment, it shouldn't be surprising that the questions that I seek to help my patients answer are very similar to Dr. Hartwell's. We seek to help our patients answer the question, do I have cancer? What stage is my cancer? That is, how dispersed is it? Is there a way through better characterizing this that I can increase the likelihood of a good outcome? And is my cancer responding to therapy? So in helping the patient and helping you understand how we do that, I have the following objectives for the next 25 minutes. The objectives are present an overview of anatomic and molecular imaging. One part of that is early diagnosis. How do we get early diagnosis? We make it so that we can see tumors early at a small size. And there's been a big shift in our ability to do that. The second is a transition from imaging, from really looking at structure, a structure that doesn't belong, which is fundamentally what cancer is, to the physiology of that structure. What's its metabolism? Because there may be a structure there. You may completely treat it and it's replaced by scar, but you won't know that if you just look at its structure. You also want to be able to understand its physiology, and that's broadly characterized as molecular imaging. Prior to 1973, if you had cancer, you would get, one of the studies you would get would be an X-ray. An X-ray is made by projecting an X-ray beam at an object and getting something a little bit like that finger puppet that's created on the wall. It gives you a very coarse delineation of anatomy, which is what you see here, such that you see dense bones, you see the skull, you see the sinuses. The things that you could see in terms of a tumor for early detection would be the presence of calcium or the presence of bony erosion. But those are very atypical with brain cancer, so it was not an effective tool. In 1973, there was the introduction of CT, which is this image set here. The difference between an X-ray, plain film, and a CT is that you rotate the X-ray beam all the way around the structure, and then you can reconstruct it. And you can make an image that's based on density alone. But what you see here is there's a great deal better delineation of the density. Here's the skull all the way around, and we've made a slice right through the mid portion of this person's skull, okay? Here's tissue, here's a fluid space, here's a fluid space. And if you divided a line right down the middle, the way that you all will help me detect where this cancer is, okay? Here's the audience participation piece, is you would say, is there asymmetry there? Does the right side look different than the left side? And hopefully, and I'm seeing some heads nodding, is you're all focusing on this area right here. Okay, it's different, it's a structure that doesn't belong, and that's how cancer detection is done with CT. It's detecting asymmetry. Now here's another quiz case for you, all right? CT, is there a tumor? Yes or no? How many say yes? How many say no? The art of medical school. I wouldn't ask you if there wasn't one there. Answer's yes. And so then the question is where is that? And in fact, it's hiding right down here, okay? And what you can see is here's the tumor, okay? The brain, this tissue here, is shifted over to the side, and there's some bigger fluid spaces. Now, arguably, that's tough to see. For me, that's job security, okay? The second part of that, however, is that what CT allows you to create your image on is density alone. So there's not as much information as you would like to really make that lesion conspicuous. We call that conspicuity. MRI changed that. With MRI, you don't just look at density. You put a person in a magnetic tube, if you will. And all the protons in your body's largely made up of water, starts to precess, and they become little magnets. And if you send in a radio wave, what you'll get back is a radio wave that's slightly modified. And you can use that to create a picture. And then you can manipulate the picture so that it shows you very nicely the presence of the disease. So if I ask you on this one, is there cancer there? You'd say yes before you even looked at it, because that's the art of taking a test. And then you would see that there's in fact a cancer there, very nicely delineated. As we get better at increasing conspicuity, again, we can start to see smaller lesions, we can pick up cancer earlier, and our likelihood of a successful intervention increases. So now, this is the previous CT I showed you. Here's an MRI, and we use a technique to make it more conspicuous. We can suppress signal from everything except what enhances after we give an intravenous agent. So, asymmetry there, yes or no? The answer is yes, there is, and it's which side? Patient's left, patient's right. I mean, your left or your right? Exactly right. And here it is. Here's another example of a brain tumor, okay? Simple as that. Now, in addition to advancing the technology that gives us increased conspicuity in terms of acquisition of information, we also can start to incorporate computers in terms of image processing. So if I said to you here, is there a tumor there? I think you'd all say absolutely. Asymmetry is bad, and this is clearly asymmetry, and that's a glioma within the brain. Now, what is very important in managing patients, though, is not just picking up the first lesion, but it's the second, it's the third, that's staging. And sometimes that can be difficult when the second lesions are small. So what we've incorporated now is computer-aided detection. You can make the lesion more conspicuous. Here you see the first lesion, but you can make it so that the second lesion is also well demonstrated. So in terms of pushing the envelope back, in terms of diagnosing cancer early, it's advancing the image acquisition devices to see things better and smaller, and then it's bringing in computer-aided detection. Now, one of the advances that comes with the ability to see things smaller is not only earlier detection of cancer, but also being able to have a safer intervention. Because if you're going to remove the tumor, in the brain, there's a lot of really important structures that live there. Your brain drives your ability to speak, to understand, to move your extremities. And so what you see here it's called a sagittal MR. Essentially, it's a profile of the brain. Here's the patient's nose here, mouth, back of the head. And what you see is some gross morphologic structures fairly well defined. But what we can do now is we can actually delineate the fiber tracks, the wires that connect the brain to the arms and the legs, the ability to speak, the ability to understand. And once you can start to define those, then what you can do is you can use that ability to say, where are the most important tracks and what's their relationship to the tumor? So what this picture shows you is it shows you first a map of the corticospinal tract. That's the track you need to be able to continue to have motor function. And here's a large tumor in green. We used a computer to make it obvious, okay? And so if one is going to try to remove the tumor, you at best want to make sure you avoid the crucial fiber tracks. So again, using imaging for increased conspicuity, better outcome. Now, there's been a shift to also start looking at not only the anatomy, but also the physiology. And the physiology is referred to as something called molecular imaging, defined as the remote imaging of a physiologic process in vivo. That's a big word. A way to think about that, and it's a little bit harsh, but my family's here, I come from a farm, is if you're driving down the road, okay, and you see a muskrat on the side of the road, muskrat's not moving, you don't know. Maybe he's with us, maybe he's not. Next day you drive by, muskrat's still there, not moving, size hasn't changed. Next day, day after. After about 2 weeks, nature starts to set in, okay? Muskrat changes size. That is largely how we've approached anatomic imaging and looking at tumor progression. You wait until it changes size. But wouldn't it be better if you could actually look and see, is the muskrat actually using oxygen? Is he using glucose? Is he starting to have cellular breakdown? That's what molecular imaging allows us to do. It not only allows us to see a structure, but see the function of that structure. And it's transforming the way that we do things. So we can now look at oxygen utilization. We can look at the measurement of apoptosis, which is programmed cell death. After the cell, at the very earliest moment, starts to die due to an intervention, you can see a change in the cell membrane. You can also look at glucose utilization, and now we're developing and implementing techniques to actually look at the replication of DNA with imaging. So let's first talk about looking at oxygen utilization. It's referred to as the BOLD effect. It stands for blood oxygen level dependence imaging. As we talked about before, when you put a patient in a magnetic field for MRI, all of the protons start to spin around, okay? And you send in a radio wave, and when that wave comes out, you can create an image. The only thing that will confound that is if that magnetic field isn't homogeneous, okay? Well, in our brains, there's a couple things that cause inhomogeneity. If you have calcium, strong amounts of calcium, blood will do it, and actually unoxygenated hemoglobin. So there's a molecule that you know that carries blood, and if oxygen is not attached to it, then it causes a little magnetic field. And what that will do is it will cause signal to go away, and you won't get as bright a picture back in your brain, okay? Now let's say a part of your brain you start looking at pictures. I look at that bright light, it starts flashing. Well, a part of my brain needs more oxygen to do that, a very focused part. And I can actually image the part of the brain that's using more oxygen because I get a little bit more signal back. The magnetic field is more homogeneous, okay? So let's take a look at that. This is a picture of a patient who's looking at a flashing light, okay? And what you can see is certain parts of the brain give back more signal. And it's these areas that are associated with vision. And you can do a similar map for language, speech, hearing, motor function. Why is that relevant? It's similar to the map I showed you of fiber tracts. It doesn't necessarily help you detect that tumor earlier, in this case, but it helps you see where are the crucial functions so that when you're trying to remove the tumor, you preserve them. I gave you the analogy about how we currently monitor effectiveness of therapy. We have based it largely on anatomic imaging. I gave you that somewhat difficult analogy, but in fact, we talk about imaging and cost, and I do think it's relevant. Imaging is the second fastest driver of cost in medicine, behind pharmaceuticals. But when we think about imaging, it needs to be thought of in the context of all of the management of the patient. So if I'm treating a patient for 4 weeks with a chemotherapeutic, it's the wrong chemotherapeutic. The patient develops complications. They get more ill. There's a delay in starting the right chemotherapeutic. There's an enormous cost to that. So one of the things we focus on is imaging done correctly, looking at the right markers, can actually take costs out of medicine if it's done right. If you target your therapy in an individualized way based on the biology. So let's talk about an example of that. This is an example of a patient with a medulloblastoma. This is a sagittal image. Here's the nose, there's the back. Here's this large enhancing tumor. It's a highly malignant tumor. It's the most common intracranial tumor that children get. Median age of onset is 5 years old. There's a number of treatment regimens that have been used, but only a subset of the patients benefit from chemotherapy, and yet all of the treated patients are exposed to the potential morbidity of the treatment. The current paradigm for assessing effectiveness, as we talked about, is somewhat limited. Ideal would be the ability to monitor changes in physiology. What would you like to be able to do? Well, rather than look at this at a morphologic level, at a structure level, you'd like to be able to do what Dr. Hartwell talked about. You'd like to be able to look in the serum and see surface cell markers. But you also, sometimes there's a problem with specificity. Where are those actually coming from? So one of the nice benefits of linking molecular imaging and molecular diagnosis is it can increase your specificity. But here's an example again. If you watch this tumor, what you'd like to be able to do is look at a cellular level. These are cells of the medulloblastoma. Well, it turns out you can do that. One of the things that starts to happen when you have the right chemotherapeutic agent is there's a change in the cell membrane. This is a depiction of the cell membrane. These are phospholipids, and on the inside of the cell, you've got a specific phospholipid called phosphatidylserine, okay? It's not on the outside, it's only on the inside. Well, the second that cell starts to die, a few of these move to the outside of the cell, okay? They're now on the outside. And what I can do is then I can send in an antibody that will actually attach to those phosphatidylserines. And I can tag that with a magnetic particle that actually causes that magnetic field inhomogeneity that I talked about. And if I can do that, then I can image it. And here's just an electron micrograph of the cell surface and some of these particles starting to attach. Here's one, there's one, there's one. And they attach to the surface of the cell at the earliest possible time. And then I can image it. And so what you see here is an example of a treated patient, and you can see how this signal is less than this, okay? And the reason is because those little particles cause me to lose signal on MR, and I can tell, even though the size of these tumors is exactly the same, this one's starting to undergo cell death. I'm doing the right thing. Apoptosis is occurring. Now that's with MR. Now another technology that there's a great deal of expertise here at the University of Washington is called positron emission tomography, or PET imaging. Okay, it's not small animals, it's PET, P-E-T. Okay, with that, rather than shining an X-ray at something, okay, or sending in a radio wave, the energy actually comes from inside of the patient. and you can detect it. What you do is you inject a radioisotope, okay, and it emits a positron. Those positrons travel a very short distance and they hit an electron, and then they give off 2 photons, okay, which photons are just, it's a beam of light, okay? And then I can put them in something that's shaped just like a CT, and it'll detect those photons, and it can tell where they came from. And the thing that's neat about it is that I can tag those positrons to an analog of glucose. So I can then give this, and it'll go to where glucose is being used the most. So if I've got tumor versus scar and I can't tell if it's still alive, I can see how much glucose goes to it, okay? I can tag it and I can see how much oxygen goes to it. I can tag it and I can see how quickly it's making cells, very early before there's any change in size. Here's an example. On your right is a glucose molecule, okay? What I can do is I'm fortunate that I have great radiochemists here. And what I can do is I can just change one of the molecules on this. This is an oxygen and a hydrogen. I can put a fluorine on it. And it'll still go where glucose goes, but you know what? I can make it emit positrons, and then I can track it, okay? And so when I look at tumor response, one of the challenges in the brain is complete tumor response to treatment may result in the tumor shrinking, but it might result in a scar. And so when I image that patient, I won't know whether it's a scar, or again, I won't know whether there's viable tumor. So here's an example actually of that being imaged. This is a patient after they've been treated. I've used MRI, and you can see there's still a lot of enhancement. And I'm not sure if that's scar, treated it right, done, or actually if it's still a viable tumor. So what I can do is I can create one of these glucose analogs, administer it to the patient, and if I see signal coming back from that glucose utilization, scars don't use glucose, tumors do. And so I can see that this is remaining tumor and the patient needs more treatment. I'll give you another example. There's something called fluoromysoisonidazole, okay, long term. Suffice it to say that on a cellular level, there's something called hypoxia-inducible factor. It's one of the proteins that can get transcribed. And in the setting of an aggressive tumor, this upregulates, okay? And what it does is it creates a local area where there's a greater degree of hypoxia. That hypoxia actually makes that tumor harder to treat. So if I can find the places in the brain where there's a tumor that has a greater amount of hypoxia, it will cause me to change my therapy. And so here's an example of that. Here's a patient who's the same patient, has a tumor here. And what I want to know is, when I talk to my radiation oncologist, how much do you hit this with radiotherapy? Well, one of the things would be maybe give it a boost in the areas where there's a greater amount of hypoxia, and then let's look at it again and see how do we do at decreasing the areas of hypoxia. Because if I decrease the areas of hypoxia, that means that my treatment is working. So here's a patient undergoing therapy. Here is pre-treatment, and you can see the areas of hypoxia in yellow, okay? Then after treatment, we re-image them, and the areas of yellow progressed. This patient did very poorly. The therapy was ineffective, and we were able to track that. Again, molecular imaging. One of the things that we've talked about here about academic universities and why do they exist, it's the innovation cycle, right? It's seeing difficult problems, tumors that you can't measure that don't respond, and then going actually and doing the research to figure out how can I get a better look at these, and then cycling it back down through teaching to clinical application. Seeing where those clinical applications don't hit the mark, and then bringing it back up to discovery. And so one of the things that we've worked on to a great degree here is what else do we need to do? Well, one of the things we were interested in is let's see if we can create a marker that looks at how quickly DNA is reproduced. Because what do tumors do? It's unregulated cell division and DNA replication. So if you could actually measure that, you'd have something, I think, very useful. And so we looked at an approach to try to come up with a way to look at that. And what we did is one of the scientists here, a gentleman named John Grierson, actually looked at thymidine, which is one of the backbones of DNA, and he said, you know what I can do? I can attach a fluorine to that. And if I attach a fluorine to that, what does that become? Audience participation? Exactly, a positron emitter. If it emits positrons, it'll go to where the greatest amount of DNA replication is, and I can see DNA replication. And so here's a study in a mouse. We start in an animal model, we create the molecule, then we develop it, make sure it's safe and not toxic, and then you go through the FDA approval process, and then you actually can implement it in treating patients. And so now we actually have incorporated this paper published in Nature into how we look at cancer. This is a patient with lung cancer, and we're able to look at the replication of that lung cancer. And then we'll use that to see, are we effective in our therapy or not? If we're not, we change therapy. It also helps us with detecting early recurrence and differentiating scar from tumor. Another example, pre-treatment, FDG, that's the glucose, goes away post-treatment, and this is the FLT. So we really have taken this through the innovation cycle to give us something that's probably even better than FDG at looking at tumors. Closing comments, fusing MRI with PET imaging, one of the things we talked about, great anatomy, great physiology, but one of the things that we're doing now is merging them. And so instead of having to get another acquisition, you can use computers, and this is an example of a PET image on top. Doesn't give you as good a picture of the anatomy, but shows you really great function. And then here's a mouse MR, okay? And you can actually map the physiologic and the anatomic information. And that's now being implemented in the clinic, both for PET-CT, PET-MR. So in the context of individualized therapy, one of the things that I think tried to emphasize is that all tumors aren't created equal. Even though it looks the same, it may not respond the same. So by being able to actually look at specific characteristics of that tumor, how it utilizes glucose in response to intervention, what happens when is apoptosis induced, what about hypoxia, you actually can individualize therapy. And although imaging is an introducer of cost, again, I would make the case that done correctly, you avoid morbidity, mortality, and you create a very effective intervention. So I think as we go off into the future, it's really going to be figuring out the interplay of using genomics, molecular diagnostics to set what's the pretest probability and what's the optimal way to make that diagnosis. In conclusion, greater and greater emphasis on prevention and early detection is really the key. Genetic screening, molecular diagnostics hold great promise. Anatomic imaging and physiologic imaging provide essential components to the goal of lessening the impact of cancer. Thank you.